Hello everyone, welcome back to the channel Microbial World. Today we will be discussing about the factors affecting microbial growth. So the microbial growth, it depends on several environmental factors. So it is not only the nutritional components, but these microorganisms, they are also depending on many environmental factors for their growth. And these factors, they are playing an important role in understanding the growth pattern of a microbe. And what are these factors? These factors include osmolarity, pH, temperature, oxygen concentration, carbon dioxide, radiation, moisture, pressure, light, mechanical and sonic stress. So the first factor we are going to discuss is osmolarity. The cells, it will be subjected to changes in osmotic pressure. What is osmotic pressure? Osmotic pressure means it is the pressure that is exerted when the solvent molecules move through a semi-permeable membrane, that is the plasma membrane. Okay. So when the solvent molecules move through a semi-permeable membrane from a region of low solute concentration to a region of high solute concentration, a pressure is exerted, and that is called osmotic pressure. So as the plasma membrane of the cell, it is freely permeable to water, what happens? There will be changes in osmotic pressure in the cell. So the water, it will move in a direction in such a way that it tries to equilibrate the cell's solute concentration to the solute concentration of the surrounding environment. Now there is two conditions. There is a hypotonic environment and hypertonic environment. Now you can see in the figure, in case of hypotonic condition, if the solute concentration of the environment it is lower than the solute concentration found inside the cell, then the environment it is said to be hypotonic. And in this case, the water it will move into the cell and the cell it will swell, and that will increase the internal pressure. And if that situation it is not rectified, then what happens? The cell it will burst from lysis of the plasma membrane. Then there is hypertonic condition. If the solute concentration of the environment it is higher than the solute concentration found inside the cell, then the environment it is said to be hypertonic. And in this case, you can see in the figure, the water it will leave the cell and the cell it will dehydrate. And extended periods of this dehydration, that will also cause permanent damage to the plasma membrane. So the cells in a hypotonic solution, they need to reduce the osmotic concentration of their cytoplasm. So these cells, they make use of mechanosensitive channels, and these mechanosensitive channels, it is located in the plasma membrane. Now how these mechanosensitive channels work? So when there is increased pressure on the plasma membrane it will stretch. Okay, and the mechanosensitive channels it will open. When this plasma membrane stretches, these mechanosensitive channels will open, and the solutes, this will allow the solutes to leave the cell, and that will decrease the osmotic pressure. Now the cells in a hypertonic solution, they need to increase the osmotic concentration of the cytoplasm. So what they do is that they will take up additional solutes from the environment. Now the cells, they have to be very careful what solutes they are taking up, because some of the solutes that can interfere with the cellular function and the metabolism and all. So these cells, they only take up compatible solutes like sugars or amino acids. They need to take up compatible solutes like sugars and amino acids, so that it does not interfere with the cellular processes. Now there are halophiles. Halophiles means they are microorganisms that have evolved to extreme hypertonic environment. Specifically, they can tolerate high salt concentration. They require the presence of high level of sodium chloride to grow. Now these halophiles, they require a sodium chloride concentration above 0.2 molarity. And they also take in both potassium and chloride ions in order to balance the effect of the hypertonic environment that they are living in. And also another thing is that all the cellular components of the halophiles like ribosomes, enzymes, transport proteins, their cell wall, plasma membrane, All these cellular components also require the presence of high concentration of both potassium and chloride ions to function. Okay. So the next factor is pH. The pH, it is defined as the negative logarithm of hydrogen ion concentration of a solution, and it is expressed in molarity. Okay. The pH scale, it is ranging from 0 to 14. 0 is representing an extremely acidic solution and 14 is representing an extremely alkaline solution. Now, on the basis of pH, the organisms, they are classified as acidophiles, neutrophiles and alkaliphiles. So, now most of the cells, they are preferring a pH that is similar to their internal environment, with their cytoplasm having a pH of 7.2 and all. So, most of the microbes, they are neutrophiles. That means they are neutral lovers. Their pH, it is in the range of 5.5 to 8. Okay. Then there is acidophiles, that is acid lovers. Their pH range, it is 0 to 5.5. So these acidophiles, they are using many kind of mechanisms in order to maintain their internal pH in an acceptable range and to preserve the stability of their plasma membrane. So these organisms, what they do is that they will transport cations like potassium ions inside the cell, that is into the cell, and they will decrease H plus movement into the cell. And they also utilize proton pumps that will pump the H plus outside. Okay. Then there is alkaliphiles, that is alkaline lovers. Their environmental pH range is 8 to 11.5. So these organisms, they need to pump protons inside in order to maintain the pH of their cytoplasm. Okay, so they make use of antiporters that will pump protons inside and sodium ions outside. Temperature. So the microorganisms, they are not having any kind of mechanism to regulate their internal temperature. And their only way is that they can adapt to the environment that they are living in. Okay, and when there is a change in temperature, that can affect the enzymes of these microorganisms, it can affect their activity and all. And every microorganism, it is having an optimum temperature. Okay, and this optimal temperature, that will result in faster metabolism and growth rate. Whereas if the temperature, if it is below the optimal temperature, that will decrease the enzyme activity, it will slow down the metabolism. Whereas if the temperature is too much high, okay, then these higher temperatures, that can denature the proteins like enzymes and carrier proteins, and that can finally lead to cell death. So the microbes, they are having a growth curve related to temperature. So you can see that growth curve. Okay. So there is an optimal temperature where the growth rate is peaks. Okay, you can see there the growth rate increases. Then there is minimum and maximum temperature. So at these temperatures, the growth will continue, but the growth, it does not happen as much as in the optimal temperature, right? And for a bacterium, the growth range is usually around 30 degrees. Based on temperature, the microorganisms, they can be grouped as psychrophiles, psychrotrophs, mesophiles, thermophiles and hyperthermophiles. Okay. So these psychrophiles, they are cold lovers. Their optimum temperature is 15 degrees Celsius or lower, and their growth range is minus 20 degrees Celsius to 20 degrees Celsius. So these microorganisms, they are mostly found in the oceans. Okay, because there the temperature is colder, right? So they are mostly found in the oceans. And how these microorganisms are adapting to the cold? They are having specific proteins are present, particularly enzymes that can function in low temperature. Okay, and they also modify their plasma membrane to keep it semi-fluid. And these psychrophiles, they are also having increased amount of unsaturated and short chain fatty acids. They also produce cryoprotectants, that is special proteins or sugars that will prevent the development of ice crystals that can damage the cell. Okay. Then there is psychrotrophs. The psychrotrophs, they are cold tolerant microbes. Their growth range is 0 to 35 degrees Celsius and their optimum temperature is 16 degrees Celsius or higher. Then mesophiles. Mesophiles, their growth optimum is 37 degrees Celsius and their growth range is 20 to 45 degrees Celsius. And almost all of the human microflora comes under this category, and also all the human pathogens. So these mesophiles, they are found in the same environment that the humans live in, okay, like in the foods that we eat, the surfaces that we touch, the water that we drink. So in all these environments, the mesophiles can be found. The thermophiles, they are heat lovers, okay. The thermophiles, they like high temperatures, and their growth range is 45 to 80 degrees Celsius, and their optimum is 60 degrees Celsius. Then there is hyperthermophiles, and their growth optimum is 88 to 106 degrees Celsius, okay. Their minimum temperature is 65 degrees Celsius and maximum temperature is 120 degrees Celsius. So both these thermophiles and hyperthermophiles, they require specialized heat stable enzymes. And these heat stable enzymes, they are resistant to denaturation and unfolding. And that is because of the presence of protective proteins known as chaperone proteins. And the plasma membrane of these organisms contain more saturated fatty acids with increased melting points. So there you can see the graph showing the temperature range and the rate of microbial growth, okay, of cyclophiles, cyclotrophs, mesophiles, thermophiles and hyperthermophiles. The next factor is oxygen concentration. So the oxygen requirement of an organism, that is linked to the type of metabolism that they are using, okay. The energy generation, that is linked to the movement of electron through the electron transport chain, where the final electron acceptor will be either oxygen or a non-oxygen molecule. Now if the organisms, they are using oxygen as the final electron acceptor, then they are engaging in aerobic respiration. So there are obligate aerobes. These obligate aerobes, they require the presence of atmospheric oxygen, that is 20% for their metabolism. Then there is microaerophiles. The microaerophiles, they require oxygen, but they require oxygen only at a lower level than the normal atmospheric levels. Okay, they can grow at levels of 2 to 10%. Then there is organisms that can grow in the absence of oxygen. They are called anaerobes. So among these anaerobes, there are different categories like facultative anaerobes, aerotolerant anaerobes and obligate anaerobes. The facultative anaerobes, they are able to grow in the presence or absence of oxygen. They just switch their metabolism to match the environment. But mostly they prefer to grow in the presence of oxygen. This is because aerobic respiration will generate large amount of energy and that will allow for faster growth. So as a result, they prefer to grow in the presence of oxygen. Then there are aerotolerant anaerobes. The aerotolerant anaerobes, they can also grow in the presence or absence of oxygen, but they are not showing any kind of preference. Then obligate anaerobes, they are strict microorganisms. They can only grow in the absence of oxygen, and they find an oxygenated environment to be toxic. Then there are oxygen byproducts, that is called reactive oxygen species or ROS. Okay, so these oxygen byproducts, they are toxic to the cells. They are even toxic to the cells using aerobic respiration. These oxygen byproducts, it includes superoxide anions, hydrogen peroxide and hydroxyl radicals. There are certain enzymes that is offering protection against these reactive oxygen species. These enzymes include superoxide dismutase. Superoxide dismutase, it breaks down superoxide radicals. Then there is catalase enzyme which breaks down hydrogen peroxide. So the obligate anaerobes, they are lacking both these enzymes. So they are not having any protection against reactive oxygen species. So you can see in the figure there is obligate aerobe. We said they require the presence of atmospheric oxygen to grow, right? So these microbes, it is found at the surface of the test tube where oxygen is available. Then there is facultative anaerobe. They are showing, we said they can grow in the presence or absence of oxygen, but they are mostly preferring to grow in the presence of oxygen. That is why most of the microbes is found at the surface of the tube. But as they can also grow in the absence of oxygen, the microbes is also distributed at the rest of the part of the test tube also. Then there is aerotolerant anaerobe, where they can also grow in the presence or absence of oxygen, but they are not showing any kind of preference. That is why the microbes, it is distributed throughout that test tube. Then there is obligate anaerobes. These microbes, they can only grow in the absence of oxygen. Okay, so they are found at the bottom of the test tube where there is no oxygen available. Then microaerophiles, we said they require oxygen, but they need oxygen only at lower levels, right? Only 2 to 10% oxygen is needed. That is why they are found growing at the below that surface portion, okay, below, just a little below the surface portion, where only little amount of oxygen will be, or lower levels of oxygen will be available. Carbon dioxide. Most of the bacteria, they require small amount of carbon dioxide for their growth. Okay, this carbon dioxide, it is either provided by the environment or it can be produced by bacteria due to cellular metabolism. Then there are capnophilic bacteria. Capnophilic bacteria, these are bacteria that require high level of carbon dioxide for their growth. Okay, an example is Brucella abortus. Brucella abortus, they require high level of carbon dioxide, that is 5 to 10% of carbon dioxide for their growth. Okay. Radiation. All the cells are susceptible to the damage that is caused by radiation. Radiation, it can adversely affect the DNA with its short wavelength and high energy. There are ionizing radiations such as X-ray and gamma rays, and they will cause mutation and destruction of the cell's DNA. Okay, the bacterial endospores, they are resistant to the harmful effects of ionizing radiation, whereas vegetative cells, they are susceptible to the impact of the radiation. Then there is a bacterium called Deinococcus radiodurans. This bacteria, even after it is exposed to radiation, it is having the ability to completely reassemble its DNA. Then ultraviolet radiation, UV radiation, that can also damage the DNA. What they do is that they will attach the thymine bases that are next to one another on the DNA strand. Okay, so they will attach the thymine bases and they form thymine dimers. These thymine dimers will inhibit DNA replication and transcription. Okay. Now, the microbes, they will be having DNA repair mechanism, okay, that will allow them to repair limited damage. Okay, one such DNA repair mechanism is enzyme photolyase that will split apart the thymine dimers. Moisture. So the water, it is an essential component for the growth of the bacteria. Okay, 80% of bacterial cell is made up of water. Desiccation or drying can have severe effect on the microbes. Okay, an example is Treponema pallidum, Neisseria gonorrhoeae. These microorganisms, they can die easily due to desiccation. Whereas bacterial pathogens like Mycobacterium tuberculosis, Staphylococcus aureus, they can survive desiccation for several weeks. So the moisture content, it is needed to maintain an optimum growth for the microorganisms. Pressure. Majority of the microbes that is living on the land or water surface, they are exposed to a pressure of approximately 1 atmosphere. Okay. There are barophiles. Barophiles, these are microorganisms that live on the bottom of the ocean, where the hydrostatic pressure can reach 600 to 1000 atmosphere. Okay. These microorganisms, they are pressure lovers. They require high pressures to survive. And these microbes, they also have increased unsaturated fatty acids in their plasma membrane and also shortened fatty acid tails. Another factor is light. Phototrophic bacteria require light for their growth. So this phototrophic bacteria, they are a group of microorganisms that capture light energy and convert it into chemical energy through the process of photosynthesis. Okay, these bacteria, they contain pigments like chlorophyll or bacteriochlorophyll that enable them to use sunlight for energy production. Most of the bacteria can grow well in darkness. Presence of ultraviolet rays and radiation can reduce bacterial growth. Photochromogenic mycobacterium, okay, it is a unique species that produce pigments only at the presence of light. Mechanical and sonic stress. The cell wall of the bacteria, it provides protection from stress-related issues. But this cell wall, it can be ruptured by vigorous shaking with glass beads or when the cells are exposed to ultrasonic vibrations and all. So this is all about the factors that affect microbial growth. Thank you.